this might be where that primary prevention is really can find a home. So consider this as an example of what could be done. Patients come in with a large aggressive tumor, they're going to get upfront chemotherapy. We know that if the upfront chemotherapy completely eliminates the tumor, it's called a pathological complete response. This is good news. Unfortunately, it doesn't happen that often. And most of these patients have residual tumor, which then comes out, blah, blah, blah. For those patients where we've had an aggressive tumor, an incomplete response to neoadjuvant therapy, can we, after the surgery, randomize them? Standard of care, chemotherapy, plus or minus this metastasis preventive. This is the experiment I want to do with the LPA1 inhibitor. I think we should be doing it with a whole bunch of inhibitors. The endpoint, metastasis-free survival, toxicity, quality of life, and about 1 million others. Okay, make sense? Made sense to me. Look at this, guys. These are FDA slides. They're publicly available. Musa Mayer sent them to me, where they're talking now about this guidance in the neoadjuvant setting. And we'll just skip down to slide 37. This pathway creates new blah, 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 metastasis. Okay? The FDA said the word metastasis endpoint. Okay. Let's go to secondary metastasis prevention, because I'm sure I'm talking too long. There was just a breakout session on it, but for those of you who weren't there, I've also been working on brain metastasis now for about 6 years, and I was very fortunate to be funded by the DoD for a Center of Excellence, huge grant for brain metastasis. This is most prevalent in the triple-negative and HER2 metastatic subpopulations. Metastases themselves and the treatments cause morbidity, neurocognitive function, seizures, on and on. Increasingly, first-cited metastatic therapy relapse, particularly in the HER2 setting. And it's increasingly causing patient deaths, okay? Traditional drug therapies don't get there. We have something called a blood-brain barrier that surrounds your brain and protects it. And the researchers have been duking it out for 20 years as to if a metastasis forms, has it broken open the blood-brain barrier? We actually did this, it took us 4 years to do it. We did the study in mice, and the answer is yes and no. There is more permeability into a brain met in a mouse, but it is not nearly as much as a metastasis in your lungs or your liver, etc. So it's about 2-fold different when it needs to be 200-fold different, okay? So these drugs aren't getting in. So this is what we've been doing. Everybody familiar with nude mice? These are mice that have a compromised immune system and just coincidentally no hair. And so you can put human tumors in them and they won't reject them. So this is a nude mouse. So we've developed a couple, 5 now, tumor cell lines that if we inject into the circulation, we go in the left heart ventricle, they will get brain mets. And so we now have an assay to look at drugs, all right? And so what we do is we inject the cells, left cardiac ventricle, day 0. For preventing the formation of brain mets, we start drug on day 3 when there's just single tumor cells in the brain. For shrinking brain mets, we start them day 21, 24, somewhere in there, okay? They're already brain mets, and we're going to ask, will this drug shrink them? Okay, here comes the endpoint. And then what we do is just section these brains and start counting. We have now tried 18 drugs in this assay. Many of the drugs that are used in breast cancer. And I'm here to tell you that only those blue arrows, there's 5 of them, had any partial preventive effect. Okay? None. 0. Shrunk establishments. There is very little role of chemotherapy in shrinking, particularly in the brain. The ones you might be in, did I have? Yes. Here are the ones that worked. I thought I would tell you. Many are experimental or approved for other cancers. Lapatinib or Tykerb is the one you know. It's a small HER2 inhibitor. It prevented metastasis formation of HER2-positive cells by about half. Okay? We've done the studies. It is more brain permeable. Vorinostat is approved for a lymphoma. It's epigenetic. It turns on different genes. It also had an effect. Pazopanib was just a cool drug. It's a Glaxo. It's approved as an anti-angiogenic in renal carcinoma. These are all brain permeable drugs. They're not the usual stuff. It has worked in 2 models. Glaxo is not going to develop it further in breast cancer because it doesn't combine with lapatinib. Okay? These are 2 other experimental drugs, PLK1, and this is actually in trial. It's a brain permeable taxane-like thing. That's it. Out of all those mice, all those experiments that we got to work. Except this. I'm presenting this for the first time, guys. We tried temozolomide. How many of you know what temozolomide is? Anybody taking it? What happened?
I did well.
Good girl. Okay. So the dose in the literature is 50 mg per kg. It means nothing to any of us because we don't dose people this way. But this is the dose all over the literature. This is the standard of care for primary brain tumors, for gliomas. So we started dosing these mice at 50 mg per kg. And we got no mets. This was the triple negative 231 line. And we thought, what happened? I mean, we had plenty of brain mets in the vehicle. So it wasn't that we missed the heart because we inject before we randomize. So we went back and we went down by half. No mets. 10, no mets. 5, no mets. It wasn't until we got to 0.5, so we're 100-fold lower than what people in the literature typically use, that we finally got, we got some mets, and at 0.5, we diluted out activity. It was a hell of a preventive effect. Would it shrink? So if we went all the way back up to 50 mg per kg, and we started at day 18, it still had some effect, but that's not where you have real mets formed. You have just the occasional brain met. If we started, I can't see this, day 24, where you have established brain metastases, it had no significant activity. It prevents, it does not treat, just like everything I've shown you. It's just life. In the glioma literature, it's known that there is an enzyme that affects whether temozolomide will be effective. It's called MGMT, don't ask me the big name. 06-methylguanine methyltransferase, blah, 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 blah. Okay. What we realized is that our cell line was dead negative for this enzyme, which would predict that these cells would be sensitive. And we really didn't know, is this the case or is this not the case? And would it be a way to stratify patients who might want to take this? So we put the gene in to the cell line. So here you can see 3 clones where we express this gene, and here was the vector transfectants, the control transfectants. And then we put them in mice. And again, I can't read this, so let's start with the vectors. Here is the vector control cells with temozolomide, no brain mets. This is a 50 mg per kg. Vector with vehicle, yep, they have brain mets. If we express NGMT in these cells, they get brain mets. In other words, the temozolomide doesn't work. So it is dependent on MGMT. What else do I have? We are writing up the paper now, and what we have done in collaboration with a group in Poland is to get 48 sets of matched primary tumors, brain metastases. These women had breast cancer. It became metastatic. They got brain mets. They chose neurosurgery, and at the same institution. Okay, so these are tough to find. And what we found is that the majority of those matched sets, the MGMT expression level in the primary tumor corresponded to those in the brain met. So that we think we can look at the patient's primary tumor and predict the expression of this gene. Here is randomized secondary brain metastasis prevention. Hang on one second, I'm coming back to you. Patients with limited brain metastases, 1 to 3, even to 5 for people I've talked with, treat it with SRS. That is the radiation just to the lesion. It's not whole brain radiation, okay? You can do that up to 10 minutes. These patients are at a very high risk for the development of more brain metastases because likely there are tumor cells sitting there that are ready to go. No whole brain radiation. They would get randomized to this metastasis preventive agent, maybe temozolomide, or the placebo in a context of standard of care, systemic care. The endpoint, again, is not shrinkage of the 1 to 3 brain mets. It is time to the development of a new brain met. Okay? Toxicity, quality of life, maybe